In PCR, a chain reaction occurs because an effect serves as a cause. So here's the effect. The effect serves as a cause. The products derived from the raising and lowering the temperature serve as the template in the next cycle, thereby increasing the number of products. Now, this sophisticated amplification technique has a number of applications, but I will describe only 2 at this time. The human genome has already been sequenced, consisting of about 3 billion DNA bases. The sequence contains genes that have various roles, such as memory and sex. But let's assume that you want to extract a partial sequence from the gene that causes cancer. Suppose this part is the gene you want to extract. Then, so you can first synthesize the short DNA primers. Short DNA primers that correspond to the terminal ends of the DNA you want. Here and here. Then you perform PCR to amplify the gene so that you obtain a large quantity of this part of DNA from the sample. You can PCR this part out. So oncogenes, or cancer-causing genes, can be identified by extracting DNA from the skin or hair of cancer patients and performing PCR. The analysis of the isolated oncogene may reveal whether the patients have developed cancer because of abnormalities in their gene. The patients can be categorized based on the analysis of their genes that are amplified or mutated. The treatment policies can be determined based on this categorization so that you can offer customized treatment. So this kind of practice is called personalized medicine. Currently, cancer is categorized by primary site, which is usually an organ, stomach, liver, or lung. There are more than 200 types of cancers. But cancer can also spread to other parts of body from the primary site. So it's not really appropriate to categorize cancers by the affected organ alone. The same drug may not work equally well for all the patients. A more accurate categorization would be achieved if based on genetic abnormalities. Then with PCR, you can isolate specific gene you are interested in. The different treatment policies can be offered for the different patients with different genetic abnormalities. Then such personalized medicine should be able to provide a better cure of the disease. So when the PCR technique became popular, the film Jurassic Park was a box office hit. In this film, the scientists found ancient mosquitoes preserved in amber. The bellies of the mosquitoes contained the blood of dinosaurs. The genes were extracted from the dinosaurs' blood and amplified with PCR. The dinosaurs were brought to life from the resulting DNA sequences. In reality, it would be hard to identify all the genes of even single dinosaurs in a drop of blood. Why? Because the DNA would have deteriorated over millions of years. However, the process that movie presented might be achieved by PCR technique if we obtain clean samples and good reasons to do so. So I'll tell you another example of PCR application, the checking the presence of genes. So let's assume that one of you is infected with bacteria. I will take a small amount of saliva from each of you and perform PCR. By using primers specific to bacteria DNA, the DNA of bacteria will be amplified by PCR if the organism is present in the saliva. If the bacteria are not there, the DNA won't be amplified. The presence of a small amount of DNA can be determined by checking whether DNA is amplified by PCR. So this method can also be used in cancer diagnosis. In a certain type of leukemia, different genes fuse and promote the proliferation of cells. In particular leukemia, BCR gene is fused with ABL gene. So then usually they are separated, but in certain type of leukemia, these two genes are fused to each other. The PCR, because genes are fused, PCR is performed using a primer that is designed to amplify the junction where the genes meet. If you do the PCR, if you get gene amplified, so then you can tell whether the gene is present or not. So PCR offers such a highly sensitive method which is applicable to the early detection of viruses and other pathogens. It is up to your imagination how you use PCR. You can do many different experiments by using PCR. The PCR is an excellent idea which allowed many different technologies. So what is the key element of PCR? I'll tell you an answer in the next video clip.